Thanks for the organizer to have invited me. We turn to the nice world of planes. Don't escape, it's only for 10 minutes. So it's going to be rather technical, and you can consider it as an application note of what was just said before, because we have more or less the same considerations. So why working on planes? I'm especially working on seats. It's a major issue to understand seed reserve accumulation for yield and nutrition values, industrial values, and we aim at gaining a better understanding of limiting factor in seed reserve accumulations. This is an embryo of Arabidopsis, 9 days after pollination. Arabidopsis is our species of interest. It's a model species, which genome is completely known and sequenced. It's our fly. The seed is more or less half a millimeter, so the embryo is very small. We know genes are specifically expressed in the L1 layer, in the epidermis. We know expression of some genes vary depending on the genetic background. And we would like to make a transcriptomic analysis of the epidermis as compared to the cortex in blue, epidermis in red, in very small samples. There aren't so many techniques to perform such an analysis, so you may think about the intact system, which was published rather recently. So technically, it's a GFP that tags the nuclei, and you purify the nuclei either by fax or with magnetic beads, but you need transgenic planes. Or you may think about laser-assisted microdissection, which was our case, because we have a platform which is equipped with a Zeiss microbeam. The outline of the talk will be rapidly some optimizing of the cytology procedures, RNA-seq procedures, and we'll present you today a kind of proof of concept and limitation we got on extremely low samples. So we are working on calyx. So calyx is what you see in rapeseed fields. It contains the seeds. This is more or less half a centimeter in length, with basic acid-acidic ethanol fixation, acetic acid. It's embedded in paraffin. Only an incubation step has been slightly modified. We make sections. And our main problem, and this for years, is the half-time of the slide. So after 5 to 10 minutes, RNA are just degrading when the slide is rehydrated. So it really makes troubles. We use pen slide, plastic covered, an RNA-free room, everything is standard. We tried 2 kits for RNA extraction, and we are using now the Arcturus PicoPure, which is the best in our hands for the best quality. Okay, the quality point. So when you leave your slides on a long time on the microscope, your quality decreases, which is rather normal. We indeed tried to perform a library with a relatively low quality RNA, and we failed. So we turned to this quality trouble and tested a few things. The quality according to microdissected surface. So to our great surprise, we have a very high variability with low surface microdissected. We thought we would have increased the quality, and it stabilizes after a certain quantity. The quality stabilizes according to concentration. It's more or less the same, of course. The concentration increases according to surface. And total RNA quantity increases according to surface, of course. But the main point is low surface allows higher speed first, and low surface produces less RNA. So we need to decrease the quantity needed for RNA-seq experiments to increase the speed and to increase the sensitivity. So we have, so our running conditions are more or less 40,000 micrometers, square micrometers, that produces more or less 0.4 nanogram total RNA. And with those conditions, we got nice quality, and we processed an experiment. So the thing is, we made one single macrodissection that produces more or less 7 nanograms of total RNAs, and we performed the library with 5, 1, 0.5, and 0.1 nanogram of total input RNA with 18 amplification cycles, which is rather low for plant cytology. We used the smarter CloneTech kit. I promise I have no contacts with the previous speaker, which is the best in our hands, 18 cycles, and we have a specific step using a covaris that breaks the double-stranded DNA into fragments of more or less 250 to 300 base pairs that are highly suitable for Illumina sequencing. So it normalizes the sizes of double-stranded CDA fragments. For RNA-seq processing, we made per ends 100 sequence on an Illumina high-sec 2000, and we multiplexed the library by 3. So we expect, I'm sorry, I have problems with the device. We expect a coverage of more or less 60 million reads, which is more or less what we got here. So I don't have the 0.1 nanogram results, so the library failed, but we do have the 0.1 nanogram library. And we got a coverage of more or less 50 million reads, which is nice. How many genes do we detect? So in Arabidopsis, we more or less have 24,000 genes. We are in a developing organ, so we don't expect all the genes to be expressed, and we identify 16,000 genes more or less in the 5 and 0.1 library. A little bit less for the 0.5, and you will see during the talk the 0.5 library is always a little bit lower than the two others. How is the number of genes found in one library compared to the other one? Actually, you have 97% of the genes detected with 5 nanograms, which are detected with 0.1, and vice versa, which is extremely nice. We were extremely happy with this result. Do we have any bias concerning the quantity of read per, so the most expressed reads, actually? So when you make a cutoff of 0.5 read per million, you have more or less the same results, once again, for the 5 nanograms and 0.1 nanogram, a little bit less for the 0.5 nanograms. So the detection of identified genes in the most expressed reads is really nice as well. What about quantification of expression? Actually, when you make a Pearson correlation on counts between the 5 nanograms and the 0.1 nanogram and 0.5 nanogram, the correlation are good. It's nice. And when you compare the same comparison with the 5 nanograms to the other one with the genes which are the most expressed, the correlation is once again very nice. So to this point, we were really very confident, and we like very much the experiment. So we can detect your genes, and apparently the quantification is nice. When you plot the quantification, so you have here the log expression for the library of 5 nanograms, and here for 0.1 in blue or 0.5 in red. You see a strange thing. So for the 0.5 nanogram, the distribution is okay. It's what you expect, more or less, for low quantity RNA. And for the blue one, you see this strange curve which looks a bit broken in two populations. Actually, we don't have an explanation for this. And we went a little bit further in the analysis, and we made a plot distribution of the reads on gene bodies. Gene bodies is a virtual gene, and you plot all the reads on this virtual gene here. So you have the 5' of the gene and the 3'. So for 5 nanograms or 0.5 nanogram, you see nice distribution. The genes are completely covered. And for the 0.1 nanogram, you see this strange curve. So apparently you have less read covering the 5' region of the gene when compared to the 3' region of the gene. once again, we have no explanation. The only thing I can say is with the library made with 0.1 nanograms, we had, when the data were computed, an excess of short sequences with poly-DT or poly-DA that were cancelled by bioinformatic treatments. So this may have an impact on the curve, but once again, we are not fully sure of the strength of quantification with extremely low libraries, or libraries made with extremely low quantities. So what are the conclusions of this work? Actually, if your question is, I want to have a black and white answer of whether my gene is expressed in my very low quantity samples, it works. You can claim, or my gene is found in my epidermis or not, with a relatively good prevision and without too many errors. But if your question is really to quantify the relative expression of two genes in your sample, you need to be careful. And we think we need some more replicates to ensure the reproducibility of the quantification pipelines. And it's to test on the same line, not biological replication, but more technical replication, to check whether what we obtain with the 0.1 nanogram library is technical error or technical event, or if with a reproducible manner, we have this 5', 3' mapping problems with reads among the genes. We are currently microdissecting epidermis from embryos to answer our initial question, and we'll be soon able to answer it, whether some genes are really differentially expressed in the epidermis during embryo maturation. And for plants, perhaps it's something relatively standard for animal biology, but for plants, we have no mention in papers of such ultra-low quantification and RNA-seq approaches. And it really opens the field of many things for plant sciences, and especially for modeling, because people want to model gene networks. They want to decrease the size of the samples to have more strength in the network. To build a network for embryo development with the full embryo is okay, but if you can discriminate regions of the embryo or sub-regions, it increases the power of the approaches. And we have a demand to have very low experiments using these techniques. And finally, I would like to thank the people on the two platforms at GPB Versailles and NeuroGV Evry who made this work, especially Karisaka and Neuroborga, who made the microdissection, and Jennifer Jansuni and Céline Labrune and Salomé Balzergue, who made the libraries and the experiments. Thank you for the invitation.
Thank you very much. The talk is open for discussion. Thank you. Yes.
I was a little bit surprised about your slides where you showed that after 10 minutes of microdissection you had huge degradation problems, and when it was under 10 minutes you had not, because it sure is not an on-off process but a kinetic process. So if you have after 10 minutes degradation, you should have also before degradation. And how do you take care that this does not influence your gene expression, and shouldn't you then better optimize your procedure before that you do not have any degradation during the whole laser microdissection process?
Yeah, sure. It's a question we have for years, even for RNA extraction, for chip processing. We always had this. We had no real answer why. So lots of people have no problem of RNA degradation on the microdissector. We do have no specific explanation. We follow the procedure we discuss with people in the animal field. I don't know what happens. It may be specific for plant tissues. Our embedding, we have speed up the processes. So I gave this 10-minute window because it's more or less what you need to draw your things. Sometimes we need just 2 or 3 minutes. But for sure, if you leave your sample there for 10 minutes or 15 minutes, you see this degradation. So Of course, you're right. It's a decreasing curve. It's not black or white. But once it's rehydrated, you have degradation troubles. I have no explanation why.
Okay.
Is that degradation specific to your tissue sample, or can that also be observed in pollen, for example?
No, we have processed a lot of tissues, plant tissues, and it's always the same. It's not only for seeds or embryos. It's also the case for roots. Or the only way we don't have degradation is to work with fresh tissues or cryosections. But cryosection makes troubles if you want to really macrodissect very specific region, because you don't have the same nice quality you have with embedded sections. So some tissue you don't detect them poorly.
Thanks.
I'm very curious. I mean, these have been for us two very interesting talks, because we are looking into the possibility of entering the field. I just wonder, can you comment on something, how your methods or how this compares, for example, with the single-cell next-gen sequencing, transcriptomic sequencing that Richard Sandberg is doing at Karolinska? I mean, he has been publishing protocols for a few years now, and he's doing a little different library prep, that's my understanding, but it seems to work very well.
You mean for sample preparation or for bioinformatics treatment?
Workflow for a single-cell, I mean...
Yeah.
There be issues with amount of RNA when people are doing single-cell next-gen transcriptomics?
Plants have some specificity, so sample preparation for plants is for sure different from human or animal cells. We have this cell wall, which makes a lot of troubles. It's hard to cut. We cannot process single cells. We are always working with organs. So for preparation, we do have a lot of differences. Even for fixation, it probably takes much longer to work with plant tissues compared to animal tissues because of the cell wall. So I don't trust too much comparison between animals and plants for sample preparation. Once you get the RNA, I think we have a lot to learn between both worlds, animal and plants, because it's probably the same. And previous talk was on animals, and we share more or less the same conclusion for total RNA extraction, same kits, so probably have the same conclusions. But for sample preparation, I will be more cautious.
Maybe also, thank you. Maybe also a general question from me. Is the RNA's degradation process similar to animals? Or is it like endonucleases and stability?
No idea. I have not checked. I have made no comparison between animal and plant RNA degradation. It's probably, you probably find papers somewhere, but we are processing only plants in our platform. We are platform plant platform specific. Specific platform plants.
Okay, thank you. If there are no further questions, thank you again for the talk.